All right, so last time when we were discussing the lecture, we were looking at protein structure, and we said that motifs are common elements of secondary structure seen in many polypeptides, useful in determining the function of unknown polypeptides. And basically, when you look at the motifs themselves, motifs are common. There are a couple kinds. You could have the Rossmann fold. That's when you have a fold or a crease at the core of the nucleotide binding site, in which there's going to be a variety of proteins. You could have another type where the protein is a beta barrel, in which the beta sheet is folded around a tube. And a third type is a helix turn helix. So these are very common. They are essentially super secondary structures. And then you have that of domains. A domain, of course, is when you have protein functional units within the larger structure. Most proteins are made of multiple domains that perform different parts of protein function. If you look, here is basically the two different types of motifs that you could see. Here you have the beta. So when you look at beta alpha beta motif, here's beta structure, the alpha structure, and then the beta structure again. And then here you have helix, a turn, and then another helix. Basically, what they do is, these are important because proteins use these, especially the helix, turn helix, proteins will use them to bind to the DNA helix. Often scientists, biologists, in studying the motifs are, it's a little bit more difficult to determine the structure than it is to determine the amino acid sequence within the protein itself. And we also went on to talk about chaperones, basically proteins that are responsible in helping the protein to fold correctly. There are many proteins that do serve this particular function, and basically what we see is that these proteins are essential for the viability as well as the fundamental importance of proteins themselves. What you see is the protein shape when you look at chaperone proteins. The barrel shape in a chaperonin is from the GRO-E family, and basically the GRO-E family of chaperone proteins, as illustrated in the picture here, it is composed of two identical rings, each with 7 identical subunits, each of which has 3 distinct domains, and incorrectly to the chamber, changing it from a hydrophobic to a hydrophilic protein there. This change will allow the protein to refold, and then after just a little bit of a short time, the protein is ejected and either folded or unfolded, and the cycle can repeat itself. And then lastly, you know that protein folding, denaturation, is when you denature the protein, is when you come out of those homeostatic conditions, whether you're looking at pH, temperature, or ionic concentration of solution. Denaturation is different from dissociation. We see that a water molecule can dissociate into hydroxide and hydrogen ions. Basically, for proteins that have this quaternary structure, the subunits may be dissociated without losing their individual tertiary structure. For example, the 4 subunits in the protein hemoglobin, which is a transport protein, can dissolve into 4 individual molecules two alpha globins and a beta globin without denaturation of the folded globin protein itself. So proteins are molecules with diverse functions. They are constructed from 20 different kinds of amino acids. Protein structure can be viewed at 4 levels. Primary structure, which is the amino acid sequence. Then you have coils and sheets called the secondary structure. The three-dimensional shape called the tertiary structure. And the individual polypeptide subunits associated in quaternary structure. Different proteins often have similar structures called motifs, and can be broken down into functional domains. Proteins themselves have a narrow range of conditions in which they fold properly. Outside of that range, proteins tend to unfold, or what we call denaturation. And under some conditions, denatured proteins can refold and become functional again. That's renaturation. Our last group is looking at fatty acids and lipids. Lipids themselves are loosely defined as a group of molecules with one main chemical characteristic. And lipids are that they are insoluble in water. We use these such as storage of fat, storage fats as in animals is one kind of lipids. Oils from that of like olive oil, corn oil, coconut oil are also lipids. You also have waxes such as beeswax and earwax, and even some vitamins fall into this category. Unlike the carbohydrates, there is no fixed ratio. Lipids themselves have a high proportion of nonpolar carbon-to-hydrogen bonds, which makes these molecules very hydrophobic. There you can see the examples: fats, oils, waxes, some vitamins. And then more specifically, you have types of fats known as triglycerides. These are composed of one glycerol molecule and 3 fatty acids. The most common are even-numbered chains that include 14 to 20 carbons, and a lot of triglycerides have many carbon-to-hydrogen bonds of fats that serve in the form of long-term energy storage. So fatty acids themselves need not to be identical. Chain length does vary. You could have those that are saturated versus those that are unsaturated. You know that refers to carbon-carbon double bonds. Saturated fat, like animal fat, is solid at room temperature. So therefore, those fats have a higher melting point. They are animal origin and base. They are often solid. Unsaturated fats have one or more carbon-carbon double bonds. They have low melting point, typically liquids at room temperature, such is that of plant origin. Then you have things known as trans fats. You've probably heard of trans fats. Basically, when you think of the fast food industry, you hear of trans fats there. And basically, what you have there, trans fats, are chemically enhanced in our foods. So there you can see structural formulas. Here would be glycerol to make up a triglyceride. So here you can see glycerol, and here you can see the 3 fatty acid chains. Take notice that all these fatty acid chains are saturated. So make note in the space-filling molecule of what happens there. But when you come over to here, where you start to throw in carbon-carbon double bonds, of course, the middle fatty acid here is unsaturated, and then the one above here and below here is polyunsaturated. Look what happens to the hydrocarbon portion, or the fatty acid chains on that triglyceride. It starts to become kinked. So when you throw in those double bonds, the molecule itself becomes kinked and will start to bend. Phospholipids, major component of biological membranes. Basically, what you have there is glycerol, and then you have 2 fatty acid nonpolar tails. Glycerol itself is polar. That would make up the head region, along with a phosphate group. So the phosphate group, you have that polar head, and you have the 2 fatty acid nonpolar tails there. So if you look at the structure, There is that phosphate group, so you have choline, the phosphate group there, which has surrounding oxygen, so we have that very polar portion to the head. The glycerol is going to be the binding agent to the fatty acids there. So this is what a typical phospholipid would look like. Of course, this part here, very hydrophilic, polar in nature. Down here we have the fatty acid end, which is very hydrophobic, non-polar characteristics. If you look here, you can see in an aqueous environment how the phospholipids would arrange themselves. The non-polar portion would be hidden inside, and the polar heads would be facing the outside towards the aqueous environment. This is Michelet's concept, where lipid molecules orient with polar hydrophilic head toward water and the non-polar hydrophilic tails away. And then here again you can see the phospholipid bilayer, more complicated structure where two layers form. Hydrophilic heads point outward, hydrophobic tails point inward. This, of course, is essential to every cell membrane. We do call that the plasma membrane or cell membrane. So that's basically ends our lecture on fats and lipids. It's a small group there that you could see. What you know is triglycerides definitely are made of fatty acids linked to glycerol. Fats contain twice as many carbon-hydrogen bonds as carbohydrates, and thus they store energy efficiently. Because that carbon-to-hydrogen bond in lipids are nonpolar, they are not water-soluble and tend to aggregate together in water. Think about when you put a drop of oil into water. It will aggregate and kind of like bubble up. Phospholipids themselves replace one fatty acid with a hydrophilic phosphate group, and this allows them to spontaneously form bilayers, which are the basis of all biological membranes. So that concludes our discussion on biochemistry. We will have a little bit of a lab on this, and then we will have our test, and then we'll, of course, we will move right into cell structure function. Thank you, and have a nice day.